Hello beautiful people, welcome back to my channel once again with another vlog. So today we are going to discuss regarding our last topic from general microbiology. So today's topic is regarding the identification of bacteria, stains and IMViC reactions. Okay, so this will be our last topic from the general microbiology introduction part. Okay, so before that I would like to request you to please like, share and subscribe to my channel. Keep supporting me and let's get started with our today's vlog. So friends, today is first topic we'll discuss regarding the identification of bacteria. So here you can say why we are doing identification of bacteria, because we have to know that what will be the how you can identify the bacteria based upon their which type of the following structure, shape, their growth and everything. So that's why the identification of bacteria is also an important part. Okay, now after isolation of bacteria in pure culture from a specimen, it has to be identified. Okay, so as you all know, our previous vlog was on media cultures and culture methods. So we will identify the bacteria from a pure culture. Okay, why we will identify? Because to know which bacteria it is. So here we need to have the following studies are necessary to characterize a bacterial strain. So we need to find out which bacteria is. So we have to go through the different types of characteristics. First is morphology of bacterial colony, and second is growth in liquid media. Third is staining. Fourth is hanging drop preparation. Fifth is biochemical test. Sixth is antigenic structure. Seventh is typing methods. Eighth is pathogenicity test, and ninth is antibiotic sensitivity test. So these are the basic and the minimum characteristics we need to know before we identify one bacteria. Okay, now we'll start with first is morphology. What do you mean by morphology? It's study about their shape and structure of the bacteria. Okay, the appearance of bacterial colony on culture medium is usually characteristics. So the following are size, shape, surface, elevation, edge, opacity, color, hemolysis, that is breakdown of RBCs, and its consistency. Consistency, you will know that it will be in a liquid form, semi-solid form and solid form. So based upon these appearance of the bacterial colony, we will come to know the morphology of the bacteria. Next, going to the second characteristics, that is growth. Commonly liquid media are peptone water and nutrient broth. I have already discussed regarding the basic nutritional requirements for a bacteria to growth is peptone water and nutrient broth in my previous vlog, which is culture media and culture method. So for growth they need minimum peptone water and nutrient broth. So the bacteria appears in the following forms, that is uniform turbidity form. Second, deposit at the bottom, that will be, it means it will be settle down at the bottom just like sediment. Okay, and third is surface pellicule formation. Pellicule, some of the bacteria they will have pellicule all over their body, one or two pellicules. So from these three kinds of different types of their growth, we can identify the bacteria, and we can grow the bacteria in liquid media which contain peptone water and nutrient broth. Next, moving to the staining. Staining methods are employed to examine spheres prepared from bacterial colony or liquid culture. Staining, mainly the main purpose of the staining in the microbiology is to identify the bacteria. Okay, identify the different kinds of bacteria. Okay, so we are having some of the stains which everyone knows that we are commonly using. Everyone, I mean the only science students who belongs to this medical field, they will come to know in the microbiology we are going to use the commonly used stains are Gram stains, Albert stains and Ziehl-Neelsen stains. Okay, Ziehl-Neelsen stain is also called as acid-fast stain. So these are the three main stains by which we can also identify the bacteria. Next, moving to our fourth method, that is hanging drop method. Hanging drop method, it helps to distinguish between motile and non-motile bacteria or microorganisms. Okay, so this is regarding your hanging drop method. Next, moving to our biochemical test. IMViC reaction, biochemicals. What do you mean by bio? Means life, that is chemical means different types of reactions and interactions which happen in the life test, that is biochemical test. This biochemical test include IMViC reactions, IMViC. So friends, as you can see that small I in second I, which is in V, after V, it is nothing but we are just adding it to make it pronounced as IMViC reaction. Okay, we don't have any reaction for that. So IMViC, I stands for indole production, M stands for methyl red, that is MR, and V stands for Voges proscaver, that is VP, and C is for catalase test. So these three biochemical tests are also important in the identification of bacteria. Next, moving to antigenic structure. They are conformed by agglutination or precipitation reaction, that is antigenic structure. Next, moving to typing methods. Typing methods like bacteriophage typing and bacteriocin typing are used for intraspecies differentiation typing methods. Okay, bacteriophage is nothing but a kind of bacteria which sometimes will act as virus. That's why we are calling it as a bacteriophage typing. Next, moving to pathogenicity test. Guinea pigs pathogenicity is nothing but we are doing a trial test. Okay, so for this test, guinea pigs, rabbits and mice are mostly used for pathogenicity test. It is the causing test, how much severe cause the organism it will harm to the animals or human beings, anyone. So regarding their pathogenicity, we are using guinea pigs, rabbits and mice. Next, moving to antibiotic sensitivity. The isolated bacterium is subjected to antibiotic sensitivity test in vitro. In vitro is nothing but in laboratory. Okay, in vivo is nothing but in our body. In vitro is nothing but in the laboratory for selective appropriate antibiotic for therapeutic use. Antibiotic sensitivity is also important. We can see that which antibiotic we can use it for as a therapeutic use. Okay, so these are your identification of bacteria, what we are doing to identify a bacteria. So these are the various factors which we will go through all this test to identify a particular bacteria. Okay friends, so this is your identification of bacteria from general microbiology. I hope you like it. If you need any depth information regarding IMViC reactions and stains, please let me know. I will also make another video on this. So I hope you like this video. Please like, share and subscribe to my channel. Keep supporting me, and in my next vlog I will be discussing regarding the different types of stains. Thank you friends. Bye bye.